All right, this is the second part of chapter 3 where we're going to talk about the different cell types. And so we'll talk about prokaryotic cell and eukaryotic cells, and we'll describe the basic features of each cell type and how they're different. Remember last time we introduced cell theory. All organisms are made of one or more cells, and all life processes occur within cells. Cells themselves are alive. Anything smaller than a cell is not truly alive. And cells come from pre-existing cells. We can't make a cell, they're too complex. And then there were two basic types of cell. Prokaryotic cells, which were small, simple cells with no internal compartments, no internal membranous organelles. These are, in the fossil records, fossils of these cells go back 3 billion years or so, and most of the fossil record is only prokaryotic cells. These cells are very, very simple in structure, but they're chemically, metabolically diverse. They can do different types of chemical reactions. Eukaryotic cells, on the other hand, are large, and they're very complex with lots of internal structures like the nucleus, the mitochondria, things like that. These separate membranous compartments inside. We call them organelles. These appear late in the fossil record, maybe the last billion years or less, and all multicellular organisms, truly multicellular organisms, are eukaryotes: plants, animals, fungi, and even some microscopic organisms. Okay, so there's details. So let's kind of dig into some of these details. Okay, prokaryotic cells. All right, these are archaea, the methanogens, thermophiles, halophiles. All of those organisms don't like oxygen. And then bacteria, aerobic and anaerobic. And if you look at archaea and bacteria, you can't tell the difference. They're the same size, they have similar shapes. It's only chemically can we tell the difference. These guys all look alike, and they all basically contain the same structures, although there might be chemical differences between the two groups. So on the left, you see a cartoon image of a prokaryotic cell. On the right, it's a electron microscope photograph, and these cells are all small. Okay, most of them are less than 1 micrometer, 1 UM in diameter, and maybe 1 to 3 micrometers in length. Okay, one of the features that is most important for them is the cell wall. The cell wall is a rigid structure, mostly made out of peptidoglycan in bacteria. That's a protein with a sugar component in it, and so it's a complex material that's linked together. It makes it rigid, and it prevents water from rushing in and bursting the cell. That's osmotic lysis, and it gives the organism its shape. Okay, and I've already talked about the basic shapes: bacillus, vibrio, coccus, spiral. That shape is due to the rigid nature of the cell wall. Okay, and that's what does that. The cell wall is, again, I mentioned peptidoglycan. Here's a diagram showing the subunits. Acetylglucosamine. So the glucose is the sugar part, the amine is the protein part, and the pentapeptides hold it together. N-acetylmuramic acid. So these are the components. Peptidoglycan is cross-linked, so it's held together by these alternating sugar units and then linked by these small peptides. This is what you find in bacteria. In archaea, it's a very similar compound but slightly chemically different, pseudomurin. And even among the bacteria, there's some variations. We have gram-positive, which have a thick layer of peptidoglycan, and they're linked by something called teichoic acids. And then in gram-negative, we have a thin layer of this, no teichoic acids, and then a second outer membrane. Okay, here this is kind of a summary. but here shows in more detail. You can see that gram-positive is a thicker, more rigid layer, and so it's good for bacteria that live in the environment. Gram-negative organisms have a second membrane. This is what makes them more resistant to detergents and things like that, and disinfectants, and then a thin peptidoglycan. Okay, and so this is an important distinction in bacteria. Another feature all bacteria and archaea have is a cell membrane, a lipid bilayer just inside the peptidoglycan or pseudomurin layer. This contains proteins that serve as channels and sensory structures and things like that. It determines what goes in and what goes out of the cell. It's how the cell interacts with the environment. It's the proteins that determine the movement of things like water and stuff like that. So you have these pores with the proteins embedded in them. Again, there's lots of detail here in the book, but the membrane is found in all cells, and in all cells it controls what goes in and what goes out. Lots of the other chemical reactions that occur in the cell occur at the membrane where it interacts with the environment. Okay, so that's the two layers that you see all the time. Then you have cytoplasm, a fluid-filled area inside the cell membrane. Okay, the cytoplasm has several distinct areas in it, but no compartments. These areas look different depending upon what chemicals are present. As I mentioned, there's no compartment, but there is a region that's distinct you can find in almost all bacteria, archaea, and that's the area that contains DNA. We refer to it as the nucleoid, but there's no membrane that separates it from the rest of the cell. It's just an area of DNA, and it appears different when we stain the cell or when you look at the cell with an electron microscope picture like right here. Almost all prokaryotes have a single circular chromosome that's highly folded. Sometimes they will have additional pieces of DNA called plasmids, but not always. And so this is a big thing. The DNA controls the cell. It's replicated before the cell reproduces. And it is where RNAs are made, and those RNAs, some of them are ribosomes. These ribosomes are numerous, and they're involved with producing proteins that the cell needs. And they're directed by the DNA. All prokaryotic cells have ribosomes. Their ribosomes are made of two parts when they're inactive, the 30S and the 50S unit. And then when they're active, they're assembled into a 70S ribosome. These S numbers are Svedberg units. They refer to the ability of the ribosome to sink in a certain type of gel. You don't need to know them, but you need to know that prokaryotes have a 70S ribosome. Okay, that's really it. That's the thing. Those are the things that you find in all prokaryotic cells. Now, some cells have additional things, but for the most part, that's it. Some cells will have a slime layer around them called a capsule or a glycocalyx. These are areas of a secreted material that help them stick to surfaces and protect them from drying out or being attacked by chemicals. A glycocalyx is more, these are always outside the cell wall and cell membrane. They can be more or less organized. When they're more organized, we call them a glycocalyx or a capsule. When they're less organized, we call them a slime layer. And these are polysaccharides. And so, like when a pier gets really slippery, often that's a glycocalyx. Okay. Some cells have protein extensions, like fimbriae or pillae. Okay. These can help them stick to surfaces or transfer materials, but they don't all have them. And even the ones that do have them don't produce them all the time. So it's not something every cell has. Uh, Probably the most important of these kind of temporary or things that only some cells have is the flagella. A prokaryotic flagella is made of a single protein, mostly flagellin, that's attached to a hook and a basal body that is then anchored into the cell wall and cell membrane, and it rotates like a propeller, and this helps bacteria swim. And they can rotate these things in one direction or the other direction to help them change direction. So often they will all rotate them in the same direction, and they'll actually swim quite quickly. But then when they want to change direction, they'll rotate them in the opposite direction, and they'll tumble. And so you see this whenever bacteria want to move. Sometimes the flagella is actually underneath the the outer membrane of like a gram-negative cell, and wrapped around the cell. And this is what causes some cells to be spiral in shape. And so it causes the whole cell to rotate when the thing rotates. And so this is why a spirochete looks like a spiral, is it has this what we call an axial filament. Okay. Another thing you sometimes see are inclusions. These are chemicals that are just stored in the cytoplasm. They can be made of lots of different things. Okay, you know, that's how they store chemicals. Usually, this is just a way to store energy, but sometimes they're gas inclusions. Sometimes they're magnetic, so they help them orient. Sometimes certain prokaryotes can make resistant stages called endospores. And so they form a kind of a dehydrated, protected spore that can germinate. And they often do this when conditions are bad. So like here's anthrax. This is a bacteria that causes anthrax, Bacillus anthracus, forming an endospore. And it does this, when things are bad. And then if things get better, this thing can remain inactive for years and years and years. But if conditions get better, it can germinate. Okay, this usually happens inside the cell, and then eventually the cell ruptures. Okay, and so the DNA is protected in here. We know we can germinate endospores that are at least thousands of years old. All right, so let's summarize. Prokaryotic cells all have a cell wall, rigid cell wall, peptoglycan or pseudomurin. They all have a plasma membrane. Okay, they all have a nucleoid region with naked DNA. Okay, no membrane. Cytoplasm and a 70S ribosome. All those other things they can have sometimes. Okay, but that's it. And always small, relatively simple. Okay. Eukaryotic cells. So we're shifting gears. Eukaryotic cells, plants, animals, fungi, protists, cells that are much larger, usually at a minimum 10 microns in their smallest diameter. So they contain hundreds of times the volume of fluid. Okay, and they contain many, many internal structures. Here seen in a diagram. Here seen in an electron microscope photograph. This stuff, most of this is the nucleus with a little bit of cytoplasm around it. Okay, the nucleus of most eukaryotic cells is bigger than an entire prokaryotic cell. It contains multiple DNA molecules. It contains separate regions that are involved in producing RNAs. Okay, it has its own double membrane with protein pores that determine what goes in and out of the nucleus. Okay, it's like a compartment onto its own. Okay, things go in and out, and so it's like a controlled chamber on its own. The nucleolus makes ribosomes. Okay, ribosomes are found in all eukaryotic cells, just like all prokaryotic cells, but they're different. Okay, eukaryotic ribosomes are larger. Okay, most of them are called ADS ribosomes, and they may attach directly to a membrane or be free in the cytoplasm. Their subunits are larger, 60S and 40S. and they're abundant. Okay, eukaryotes also have a 70S ribosome like bacteria. Okay, but it's found only in organelles, in chloroplasts and mitochondria, which I'll say more about later. So they have two types of ribosomes. The 70S ribosome restricted to organelles. They also have a membrane system surrounding the nucleus, sometimes called the endomembrane system. It contains a series of channels where things are made. It may have ribosomes attached to them. We refer to it as rough endoplasmic reticulum. If it doesn't, it's smooth, and it's often involved in the production of lipids. Okay? It can sometimes send materials to a packaging system called the Golgi apparatus, where it makes other compartments like lysosomes. Okay, and so this system is really unique. Most proteins are made in the rough endoplasmic reticulum, which can be very, very extensive and closely tied to the nucleus, like shown here. They also have this packaging system so that they can make special little packages like lysosomes or perioxysomes or just vesicles to be secreted. This is the Golgi apparatus. Okay. Eukaryotes also produce an internal structural and transport system called the cytoskeleton. It's more than just a skeleton, but it's a network of several different types of proteins that are involved in giving cells shape, producing movement, controlling activities like mitosis or secretion. This network of proteins includes microtubules, microfilaments, and intermediate filaments. These things are assembled and can be disassembled. They form tractways for moving materials throughout the cell, and so you have proteins that just kind of move along these trackways. They also form structures like centrioles for movement of chromosomes, cilia for movement of things on the external surface, and the eukaryotic flagella. They're attached to the membranes of eukaryotic cells, and they can cause eukaryotic cells to engulf particles or to secrete things at their surface, so they control lots of movements. Not at all in prokaryotic cells. In addition, there are organelles that are really unique. They're separate compartments. They have specific jobs that happen only in those compartments. The most important of these is the mitochondria. It occurs in all eukaryotic cells, and it is the only place in eukaryotic cells where aerobic metabolism takes place. And so this is where oxygen is used, the enzymes to use oxygen, and this also is where ATP is produced. These things are self-replicating, and they have their own chromosomes and their own 70S ribosome. And their chromosome is a single circular chromosome, like a bacterial cell. Chloroplasts are very similar. They too have an external membrane. They too have their own chromosome. They too undergo binary fission and have a 70S ribosome, just like bacteria. And so these organelles are unique in that they can reproduce on their own. And there's a theory there that suggests that these are actually bacterial cells that have been engulfed by eukaryotic cells and now are permanent residents. And so this endosymbiont theory is an important theory on the origin of eukaryotic cells, and it explains a lot of their activities. Okay, I'm going fast. You can always go back, replay this. It's important to get an overview. Okay, and so in summary, eukaryotic cells, large, complex cells, all containing a nucleus, all containing an 80S ribosome, all having cytoskeleton and mitochondria. Okay, some have extensive endomembrane systems. Okay, some have chloroplasts. Okay, so these are the two types of cells, prokaryotic and eukaryotic, very different. Okay, all organisms fall into one category or the other.